Yes, good afternoon. Can you hear me?
Yes, sir.
Yes,
You're
sir.
Yes, sir.
audible,
You're
sir.
audible, sir. Okay, and
So,
visible also, right?
yes, sir. You're
Yes, sir.
visible,
You're
sir.
visible, sir. Okay, thank you.
Now, I
Now
welcome
I
Dr.
welcome
Falguni
Dr.
Pati,
Falguni
Associate
Pati,
Professor,
Associate
Department
Professor,
of
Department of
Biomedical
Biomedical
Engineering from
Engineering from
IIT
IIT
Hyderabad.
Hyderabad.
So, he will be
So he
giving
will be
lecture
giving
on
lecture
recent
on
trends in
recent trends
3D
in 3D
printing,
printing,
bioprinting for
bioprinting for
biomedical
biomedical
applications.
application.
Now, I
Now I
request Ms.
request
Simana
Ms.
Devi
Simana Devi
to
to
introduce our
introduce our
speaker to the
speaker to the
participants.
participants.
Over to
Over
Ms.
to Ms.
Simana Devi.
Simana Devi.
Dr. Falguni Pati is an associate professor in the Biomedical Engineering Department of IIT Hyderabad. He completed a PhD from IIT Kharagpur and had postdoctoral appointments in Postic, South Korea and KTH, Sweden prior to joining IIT Hyderabad in the year 2015. His labs at IIT Hyderabad, biofabrications and tissue engineering labs focus on development innovative tissue engineering product and in virtual tissues organ models with 3D bioprinting technology. In this area, interest includes developing novel bioinks, bioprinting methods and protocol for the production of tissues analogs on demand and in virtual tissue organ models. Dr. Falguni Pati designed and developed a course on biofabrications for the postgraduate students at IIT Hyderabad, which is the first of its kind in India. His research is supported by MHRD, DST, SERB, and DBT. He is the beneficiary of UAY grant, a new initiative from MHRD. 25% financial support is provided by an industry partner. He is a recipient of many awards, including the prestigious Ramalinga Swamy Fellowship in the year 2016. Dr. Falguni Pati has filed US patent and 5 Indian patents. He has authored over 28 journal papers and 10 book chapters, one book, and presented over 50 keynotes and invited lectures. Sir, I welcome you for the session, sir.
Thank you very much for the nice introduction, and thank you very much for giving me the opportunity to discuss and talk on our research interest. Like my this is my very research interest, that is 3D bioprinting, and in our lab we work on this every day. So welcome all of you, and a very good afternoon
Thank
to
you,
all
sir.
of you. Yeah, sorry.
Yes,
Yes, sir.
you're
Please
audible.
proceed, sir.
Okay, yeah. Thank you. So the title of this talk is 3D bioprinting for biomedical applications. So I'll be talking the recent trends in this technology, like what is the 3D bioprinting and what are the different types of 3D bioprinting technology, because this is not a single technology basically. And then what are the areas or where exactly this 3D bioprinting technology can be used. Okay, so I will. So this talk is like I kept it more general so that you all probably can be well versed with this and can well participate in this discussion. And there are very few research slides also from my work, but mostly it is general. Okay, so coming to 3D bioprinting, probably you have must have some idea about 3D printing. What is 3D printing technology? 3D printing technology is like when it is like a layer by layer manufacturing technology, starting from a CAD file to develop certain 3D structures. Okay, so in the same way, 3D bioprinting is a technology where uh, here also we print the structure in a layer by layer fashion, but here, because you will understand, because our ultimate aim is to print tissues or organs, right? So that's why if we try to print a tissue or organ, then definitely that we need to have the raw materials, that is cells, because in our body the functional units are the cells, right? So those cells, so we collect those cells, we use those cells basically. So cells can be of various types, like either it can be a primary cells taken from a patient, or it can be stem cells taken from the patient, and various types of stem cells taken from the patient. Also nowadays, this induced pluripotent stem cells, that has come in a big way, so we can also use induced pluripotent stem cells. So we use those cells, and we also use some materials like biologically relevant materials, like it can be collagen, it can be keratin, it can be decellularized extracellular matrix, so it can be anything. Like those many hydrogels, basically those can be used. So when we mix the cells plus the hydrogels and then use them for printing, so that we call bioink, that the mixture of cells plus the material, and then we use that bioink for printing tissues and organs in a layer by layer fashion with the help of a bioprinter for developing artificial tissues and organs. So that is the whole scheme, whole target of this technology and the whole process basically. So now coming to how the different steps into this technology. So very first step is the developing the CAD model. That is like in for 3D printing also we do the same thing. We develop the CAD model, but there we develop the CAD model with the help of some CAD software, like but like probably there are many softwares, some of them probably. And then we use any softwares for developing the CAD model. But in case of bioprinting, what we do, we start with the patient's CT or MRI data. Like if we get those data they get the data from the patients. So then from the CT or MRI data, we can delineate, or we can basically we can separate or can isolate the structure of our interest. Suppose we are interested in the bone printing of bone structure, then from the CT data we can isolate or we can delineate the bone structure from the CT data, and then that can be developed as a CAD model, and that can be further used for 3D printing, 3D bioprinting purposes. So in this step, pre-processing step, they actually we develop the CAD models and also the codes like for bioprinting for these things. And then the next step, that is the processing. Here actually in this process step, that is a very critical step where we do the 3D printing process. The bioprinting process is done, where we use a 3D bioprinter, like you can see here. Just let me switch on the PowerPoint. Okay, now you can see the PowerPoint, right? Okay, so where we use the 3D bioprinting, this kind of bioprinters. Okay, and what is there in the bioprinter? We need to load the materials or the bioink that is required for printing this structure, right? So for the bioink, the bioink is a mixture of the cells plus growth factors or any other biomarkers like cytokines, other things, and then hydrogels can also be there, and then some kind of scaffolding materials also we can use for printing the structure. Okay, so then this bioink is printed into a 3D structure with the help of this 3D bioprinter. So we then, what are the printed structure? That is, we call them pre-tissues because that is not matured yet, and that has not matured into a proper tissue because it is a cluster of cells plus materials. Here, this tissue is probably at the beginning, like where we have just printed that time, all the function is not yet also established. Okay, so that's why what are the printed structure? That is, we call them print tissues that needs further maturation, and then we need mature them in a bioreactor, and then they can be developed to a tissue. And then after the tissue development, then that can be used for other implantation, or that can also be used for as an in vitro tissue or organ models for drug discovery. Also, if we are using some kind of cancer cells or disease cells, then we can develop the in vitro cancer model or in vitro disease models. So that's the whole schematic of this 3D bioprinting process, where we start with the pre-processing, starting from the CT/MRI data, and then we develop the tissue, and then can be used for various purposes. Now, why you are interested in this 3D bioprinting, and why do you see now there is a tremendous amount of interest in this technology, and there are many research groups working on this area. In India also there are few research groups working in this particular field in 3D bioprinting, because there are certain advantages to this technology. Now, these are the purposes for what we are interested to this 3D bioprinting. Like, so very first example is the tissue engineering, where in tissue engineering, where we use the bioprinting, we use the cells materials to develop the artificial tissues like bioprinted skin, bioprinted cartilage, vasculature, or even liver nodules. Something like here, our interest is to develop the tissue, and that can be well used for implantation purpose. So that can be used where if the patient has any kind of trauma, injury, or any disease that due to that some tissues or organ are failing. So there we can use this. The next is like for basic understanding of this the tissue development process. So where suppose we are trying to print by a human model for human physiology and pathology. So that models can be studied for and to understand the developmental biology. Also, the in vitro disease models can be developed, so by which we can understand how the disease progress and how we can develop the disease models basically. And for cancer study, like also that is also a very good platform. Like when you can develop these in vitro cancer models, that can be used for screening drugs and molecules like that. So that help for drug discovery process. The next thing is this 3D bioprinting can also be used for in vitro diagnostic, where like you must have heard of this thing like liver on a chip, lung on a chip, or lab on a chip technologies, right? So this 3D bioprinting can also be used to discover 3-dimensional tissues or organs. So where these tissues or organs can be connected to each other by the microfluidics platform, and that can be be used for screening purposes or for better understanding purposes. So this is the overall the different purposes of 3D bioprinting. this video here, this video is a very beautiful video where it's clearly showing you how this technology actually, how this the printing technology is. Like you see, this is the cartridges or the syringes. Okay, so where we load our bioic. So these syringes, they are like here. You see, there are 4 syringes. Okay, so in 4 different syringes, we can use 4 different bioinks, or in a way 4 different types of cells we can load into the each of these bioinks. Okay, so suppose in the first syringe, one type of bioink where one type of cells are used, one type of particular cell, suppose it is endothelial cells are used, and the next suppose muscle cells are used, and the next one is stem cells are used, and then the fourth one maybe some other type of cells, other type of stem cells are used. So 4 different types of stem cells, 4 different types of materials can be used at a time. Then one material, one by one, this syringe will go down, and then the cells will be plotted according to as per our command or as per our interest, where these cells will be plotted. Suppose here these cells are plotted in a circular fashion to develop the very fast layer structure. Okay, so this is how the layers are printed in a particular fashion. So this printing pattern will be basically designed depending upon the command you are giving or the CAD model you have developed. Okay, and then you can do various other things. You can print multiple cells, you can print multiple materials, and then this process will be repeated again and again to develop the whole construct. So this is the overall scheme of bioprinting, how this process is done basically. So this process keep on repeating to the whole structure. And then you allow, if you allow them to grow, they will go into the cell number will increase, the cells will produce their extracellular matrix, and then the tissue will be matured and then can be ready for implantation purpose. So this is how the process is basically for the 3D bioprinting. Okay. Now coming to the different bioprinting approaches or bioprinting technologies, basically as I said or at the beginning, like this is not a single technology, this is an umbrella of technologies where different types of bioprinters are basically used. So there are like mostly here 3 types of bioprinting technologies, like inkjet bioprinting, extrusion-based bioprinting, and also laser-assisted bioprinting. In inkjet bioprinting, this technology is very similar to your office printer. If you have an office printer where it works on inkjet principle, then definitely there is a very similar to that. Okay, here basically we load the material in the bioink and that come to the cartridge, through the cartridge that come to the ink, so the print heads, and in the print heads there are capillaries, so that basically through the capillary you can generate the droplets by either by a heating mechanism or by a piezoelectric actuator mechanism. There's 2 types of mechanism possible, and then by that you can develop these droplets. Okay, so then all these droplets can be patterned on a stage or on a piece of paper that can be patterned as per your wish or as per the command. You can basically create the pattern with these droplets. And this here in this liquid, whatever this liquid, that may be a buffer, that may be a media. So there actually you can also load cells, okay, or any other bioactive factors. Okay, and then you can filter that so that you basically you can able to print the cells along with these droplets, and then you can do the things. In case of micro extrusion bioprinting, where basically it works on like similar to our injection syringe, where basically you load in the injection syringe, you load some drug or injectable, that is a liquid form. You load that and then you push that through by your hand, and to push that material out to the needle. It is almost very similar to the same thing, where inside the syringe we loaded bioink. So the bioink is nothing but the cell and hydrogel, and then basically now we are going to push this material out through the needle. So for that, what you need either way a pneumatic. We need a pneumatic pressure. Pneumatic is nothing but the compressed air pressure, or maybe you need the piston. Okay, or there can be a screw driven mechanism. By this way the material can be pushed through this nozzle, so that you can basically plot with this the material, you can plot the structure, whatever the structure you want, we can plot that thing. Because of laser assisted bioprinting, it is little different from the other 2 technologies, where actually we have the ribbon. So the ribbon consists of 3 layers. Basically the very first layer, the top layer, is the glass slide, and the next layer, that is the gold layer, is the energy absorbing layer, and the belowmost layer is the bioink, that is coated just below the gold layer, Okay, and then what is done? When we shine the laser on this, laser pulse on this, the laser can pass through the glass slide, right? And then it hit the gold layer. Now, when the laser hitting the gold layer, basically it is transferring energy to the gold layer. The gold will get heated up, and you know that whenever a buying is nothing but a hydrogels, or it has 90% water in it. Okay, so whenever you're hitting the gold, there will be a bubble will be created, and that bubble can dislodge the material in a drop manner, and then that material can be collected in the collecting plate. Okay, so that's the laser assisted bioprinting. It's little bit elaborate about these different technologies. Inkjet based bioprinting, as I said to you, this is nothing but a printing droplets where the cells can also be there. Suppose in this pattern, we can pattern the cells in a circular fashion over a piece of biopaper. Now, what is the biopaper and why it is important? The biopaper is made up of collagen gel, because we see with these liquid droplets we cannot make a 3D structure, right? So this will just, if you put the next droplet on top of this, if you try to do that also, then the water droplet they'll just slide away. So what do we need? We need a biopaper. So first we put a biopaper, then we put the cellular droplets here, cell droplet, and then again we put the next biopaper, and the next layer of droplets, another layer of biopaper. This same processes keep on repeating, and then we get the whole structure. Now, this biopaper is a biodegradable material, and it can degrade over time, so that you will get only the shells. They can self-assemble themselves. They can produce the extracellular matrix, and they will get the finite tissue. So this is the schematic of inkjet bioprinting. And coming to the in terms of the technology, there are two types of thing. One is the drop on demand, and another is continuous jet printing. In the DoD drop on demand technology, where you have either two types of mechanisms are possible. Either it can have a heater, and heater is the same story. If you have heater, if you are hitting the heater, then what will happen? There is a liquid column where the hydrogel is present. There will be again the bubble will be produced, and then when there is bubble produced, it is taking some volume. So small amount of liquid droplet will come out, that will be ejected. And then again, if you switch off the heater, this bubble will collapse, and then the same process. Again, it will be heated on and off, on and off. By this way, actually, you can control the generation of these droplets. In case of piezoelectric crystals, what happens if you give an electric charge to that piezoelectric crystal, it will start vibrating, right? This is a piezoelectric, that's a transducer. When that transduces or that converts the electrical energy to mechanical energy, so that is why. So in this liquid column, if something is there, piezoelectric crystal is there, and that is vibrating, then what will happen? It creates a disturbance in the liquid column, and by this way, the droplets will be generated from the bottom. Okay, so this is the little bit elaborative description about the thermal inkjet printing. When the heater is on, then the bubble will get produced. The bubbles will increase in size, and then the material will get dislodged, and again the bubble will get collapsed after some time, and then you can get the droplets after droplets. In piezo, the mechanism is different. Here, where we have a piezoelectric crystals. If you give electron signal, then the piezoelectric crystals start vibrating. And by this way, there is a disturbance, and the droplets will get generated, right? So this looks like this is a 3D bioprinter, and inkjet-based 3D bioprinter, and it looks very similar to your office printer, isn't it? So the difference is here the paper tray is being replaced by a plate, cell culture plate, where actually we try to print, or even a glass slide where you're trying to print the tissue, and the ink cartridge or the ink that has this, that is replaced by the bioink. So in the cartridge we have the bioink loaded here, and that bioink will come to the printed, and then it can be printed. So here, this is a video about the same technology, how we do the cell bioprinting with us, inkjet-based bioprinting. You see, it's not a single nozzle. There are multiple nozzles lined up in the printed. Okay, and it can be a thermal or it can be piezo. By any of such technology, basically you are generating the droplets, right? So these droplets can be printed at a certain location. Right now, here they will show you how this actually printing is done. Suppose they have taken example of this thing, skin tissue defect. Suppose there is a burn or there is an injury on the skin, there is a defect. Now you want to fill that defect or to print the tissue directly on the defect, so that you can do that by the help of this inkjet bioprinting technology, where basically these droplets can be directly printed on the wound bed. Okay, And this in the skin, you know that the skin is composed of two layers. Most basically, gross layer is composed of two layers, the dermis and epidermis. So that you can print two types of cells that would be done. So the fibroblast and keratinocytes that can be printed, and that will it is expected that this process will help to repair that wound faster and to regenerate the wound faster. Okay. Now coming to the extrusion-based bioprinting, this technology was first demonstrated by the Freiburg Materials Research Center and in Germany. So they have developed this technology around 2000, and they have commercialized this in terms of they have started a company in Tech. Now they are the major player in this 3D bioprinting, in extrusion-based 3D bioprinting, and they have this 3D bioprinter. And depending on the capabilities, there are various types of 3D bioprinters possible. I'm trying to show you the very simple 3D bioprinters that we have in our laboratory here in IIT Hyderabad. Like this is the schematic, this is the overall scheme of this extrusion-based bioprinting, where the bio-ink is loaded inside the syringe, and then it is pushed through the pneumatic or by piston. So when you're pushing, the material will come out through the nozzle, right? And then when the material is coming out, you can basically plot them in any shape, size, whatever you want. Okay, so that depending upon the geometry can be done. So this is the bioprint that we have in our laboratory here. And this is the video from our laboratory where we have printed the gelatin along with cells with the help of our 3D bioprinter. Basically, how this material is being printed in lattice structure, that is the 090 lattice structure with some porosity. You see some porosity is there in between line. With the line, there are some gaps, right? So this is the video of the bioprinting, how we can do this. So this is a very simple structure, but we can print very complex also with this thing. That only the difference is we need to have a design of the CAD model of this thing, so that by that we can able to print this. And these lines are very fine. It's almost like this nozzle size is 200 microns. So this lines also in that range, but this video is actually magnified so that you can able to see that thing. Now this is there are various components of this 3D bioprinting systems of the extrusion bioprinting. So there are motion controller system, there are the dispensing system, there are bioprinting heads, and also the computer is required for the software and other part, right? Now this video is from a Harvard Medical School, Professor Jennifer Lewis group, where they have seen how the 3D bioprinting is. Here it is clearly showing you how the bio-ink is getting printed, okay? And they have added different dyes, fluorescent dyes, so that you can be able to see the different structure and different bio-ink loaded with different types of dyes are used for printing different layers, so that this process is well visible. You see how this process is done. So now within this hydrogel, the cells are also loaded, and then many other things can be loaded. So whatever you want, based upon your interest, you can load whatever you want in this hydrogel, and that can be printed into this kind of porous dimension structures, right? So then coming to the process parameter for this extrusion-based bioprinting, these are the process parameters, and there can be many such, because any kind of 3D printing, this is not a plug-and-play technology. So you need to optimize different process parameters to print a good. So that's why. So these are all different types of process parameters, like viscosity of the material, nozzle diameter, nozzle height, pneumatic pressure, extrusion rate, print speed, gelatin kinetics. Now, why gelatin kinetics? Gelatin is a major factor for extrusion-based bioprinting, because the material, when you are printing that, the material should be able to flow. So the material should be able to come out through the nozzle, and then after printing, the material should be able to cross-link, or material should be gel, so that after printing there should not be any flow, any further flow, and you can get a very nice this kind of structure. So that's why gelatin is important, and gelatin is done by various ways. Either it can be chemical, it can be photo, or it can be ionic. These different types of gelatin kinetics can be used for bioprinting. Now, in our laboratory, we are mostly working on the extrusion-based bioprinting technology, where we work on these three aspects actually. Like one thing is the cell-laden hydrogel, where basically only the cell that is encapsulated within the hydrogel that is being printed into a 3D structure. If you see, this is the image of a cell-laden structure, where the cells, you see here the cells, the green dots are the live cells that is present within the structure. The red one are the dead cells. Okay, so this is how this cell-laden structure will look like. The next thing is the sacrificial material can also be used for certain applications, where suppose your structure is very complex, where there is some kind of hanging geometry is there. Okay, it's because in 3D printing we need to support the hanging geometry, otherwise they will just fall away and we will not get any structure, right? So that kind of sacrificial material can be well used for giving the support to the hanging geometries, and there are some kind of this bio compatible sacrificial materials are mostly used for bioprinting applications. So these are like either it can be carbohydrate glass or it can be PVA or it can be PAG. So this kind of different types of sacrificial material can be used. Also, other thing, we can also use anaplastic material, like that is basically used for FDM or FFF technique. Like where these plastic materials can also be used to make the templates, and in between the templates or in between the pores of these templates, where we can print our silicon hydrogels to get a very good structure. Like in this case, PCL framework is being used, and then in between the gap, there the osteocyte or osteoblast or chondrocytes are used to print the structure. In case of laser-assisted bioprinting, as I already told you, that the ribbon is being used here, where in the ribbon the top surface is the glass, and then below that one there is a gold layer here, that is the energy absorbing layer, and just below that there is coated. Okay, from the donor slide, if you hit the gold layer through the glass, and then there is again the same story. Whenever it's getting heated up, there will be, and then you can actually deposit the slide. Just the same thing. I have a technology where easily you can print this kind of structure. This CAD model, you have to print this kind of structure. 2 layer. Will you hear me now? Is it still breaking?
Yes, sir. Your
Is it
voice
okay now?
breaking. Your voice is breaking, sir.
Voice is breaking?
Yes, sir.
Oh, maybe. Okay, just a minute. Let me check. There is some problem.
That was internet issue, sir. Please continue, sir.
Is it fine now?
Yeah, yeah. Now.
Okay,
fine,
is
sir.
it fine now?
Yeah. Now, please continue.
Oh, okay, thank you. Yeah, I will do that. There are some maybe internet connection issue now that nowadays it is very difficult. Yeah, okay. Anyway, suppose whenever you are trying to print this kind of structures, just a minute, I can switch on the laser. Yeah. Suppose this you are able to print, you are interested to print this kind of structure where your this is the CAD model, and then with the help of this bioprinting technology, you will be able to drop these droplets and printing this. So this is how when the laser is striking on, this is bubble being produced and the droplet can be getting transferred. Now that's why this technology also called lift. Lift, why it is lift called lift? Laser induced forward transfer. Okay, so that technology is also called lift technology. And these are the process parameters for this technology. There can be the laser related parameters like laser intensity, laser spot size, laser pulse, pulse duration. All these things are possible. All these things are the laser parameters, and viscosity of the material, surface tension, cell density, and the distance between the ribbon and the collecting plate. So those things are also very much required. And that is the process parameters. And the depth of the ribbon is another process parameter that is required. Now this video will tell you how this laser assisted bioprinting process is done. Basically, so we are putting the laser pulse, right? So as the laser pulse is hitting the mirror, there's a mirror to deflect the laser pulse in the towards this lens and towards this ribbon. So whenever the laser pulse is hitting the ribbon, the story like that, the gold layer getting heated up, and then the bubble is being produced, and then the material is getting dislodged from there. And basically, as per your CAD geometry, the laser will move on the top surface of the glass slide, and the droplets will be produced, and that will be deposited on the collecting slide. So this is the process about the laser assisted bioprinting process. Now this is a very recent development where what the researchers are doing now in 3D printing. There is a major limitation is we cannot print a very low viscous material, right? If we print a very low viscous material, then we will not get a very good structure. The material will just flow away. will not be able to control the will not be able to control the architecture of the structure, or the structure the resolution will not be that great. So what the other group have started, they started printing in suspension bath or supporting bath. What is this supporting bath here? This the supporting bath is a material, or is also a gel basically, and that will support whatever you are printing, whatever the structure you are printing in that will just support. Okay, so what you do, the same extrusion based pipe printing, the nozzle can be immersed in a suspension bath and just start printing there itself. So the whatever you are printing, that will keep on floating in that suspension bath. Okay, so that you will be able to make a good structure. This is the video where from the MIT Media Lab. So there it is clearly showing you how this printing is done. Basically, you see within the supporting bath, the needle is pierced, and then it is depositing the material within the suspension bath itself. Okay, so that you can make a very good structure with this thing, and you don't have you have full freedom to print the structure in any way. Okay, basically you can create geometry, you can create a porous geometry in Z axis, and that is that is very difficult to do by any other 3D printing technique. But with this technique, it's very easy. So creating porosity in the Z, those who are working in 3D printing, you know that creating porosity in Z axis is very difficult with other technology. Okay, so in our lab also we have started doing that. Actually, here this is the thing. Like here, suppose if you have a very low viscous material and then you are trying to print that into a structure, you will not be able to do that because the material will just flow away, and that drop will get bigger and bigger. So that is the major problem for in 3D bioprinting, because the cells like low viscous material or soft material, soft gel like this. Okay, but with the soft gel you will not be able to make structure. But the same gel, if you try to print in a suspension bath, you would be able to make a very good structure. You see this gel after printing, this is not just flowing away. They stick there, they're staying wherever you have printed them, right? So that's why they are confined to that position, printing position, and that is very much required for your printing fidelity. With this technology, we have printed various structures. So here we have shown you just a three-dimensional structure, a cylindrical structure can be printed, and the beauty of this technique, the supporting bath and the material both are biocompatible. So you can also load cells into that gel, and then you can print a structure. So this is the structure we have printed with this thing. Now there are two most important properties of the suspension bath that I'd like to discuss. Like the first thing is the supporting bath should have a critical yield stress. And what is that? Because the supporting bath should not flow by its own unless you put the needle inside that, and you are moving the needle inside the bath, the material will not flow. So there is a critical yield stress. Beyond that yield stress, only the material, the supporting bath material, that the gel, that will move. The other thing is self-filling of the supporting bath material, because once you have moved your syringe, so the path, the receding path where the Z is just already moved on, so that part the gel should be able to come to this original position. So that it should be able to heal itself. So that is the self-filling property. So these are the two major important properties of this thing. And recently there is a major breakthrough in this technology, like where 3D bioprinting of human heart is done in this supporting bath, and this is from Tel Aviv University in Turkey. So what they have done in Israel, Tel Aviv University in Israel, what they have done basically, they have used the CAD model, CT data of the human heart, and then converted it into a CAD model and printed with this suspension bath. They have printed the 4-chamber heart, that is we all have, right? So the 4-chamber heart is printed with this thing, keeping the material in the suspension bath. Okay, and then they have also used the stem cells taken from the patient. Okay, so what we can actually collect the stem cells from the patient. Also, we can develop materials, and then we can mix the stem cells with the materials, and then we can print them into a suspension bath, into a 3D structure. But then the next challenge is to mature the heart tissue, right? Because as I told you, soon after printing, it will not be like a tissue itself. Okay, so this needs further maturation. So we need to mature the tissue, the heart cells, that they can beat within the structure. But their beating is not synchronous, because the signal, we have electrical signals in our heart that is actually that is directing the heart cells to beat in a synchronous manner. That's why you have this heart beating, so that the heart can pump blood out, right? But in this case, that is not present because it is not yet mature. So their next challenge is to how they can mature this heart tissue to have this function, like the breathing function. Okay, so but this is a major breakthrough where somebody has printed a human heart. Not this is not exactly the Mr. Size and shape of the human heart. This is more like a guinea pig heart, but this is a four-chamber heart with all the heart muscle, the blood vessels, all these things are present. Okay, but okay, that's a very good recent development. Now coming to the most important ingredient of this 3D bioprinting technology, that is the bioink. And this is nothing but the printable material that we use for our bioprinting applications, where this is a cell-laden hydrogel. And what is that? Basically, when you mix the cells within the hydrogel, and then you can use them for bioprinting. And this is, and probably you must have well understood, this is the most vital ingredient of the bioprinting process, because here actually this is the bioink, where the cells should be viable, they should be functional. We need to have all the conditions, like whatever is required for the cell viability function, we need to have all these things. And the hydrogel, what it used for bioprinting, they should have like all these different types of properties, and there are many more actually. But here actually I have listed 4 major properties. Those are like the biological properties, they should be biocompatible, biofunctional. So the idea is they should not kill the cells, and they should allow the cells to function, allow the cells to survive or proliferate or thrive there, right? And from rheological viewpoint, they should have a sufficient viscosity, so that whenever we are pushing the material, whenever we are actually forcing the material out, whenever printing them, that time only the material should flow. Other than the material should not flow by itself. That yield stress is required for that purpose actually. Then the other thing is the gelatin mechanism. Whatever the gelatin mechanism you are using, that should be cytocompatible, because for gelatin we can use various chemicals, right? But not all chemicals are biocompatible. They can also kill the cells, right? So that's why we need to have some cytocompatible gelatin mechanism. And the different types of biomaterials are possible nowadays, right? Like hyaluronic acid, alginate, chitosan, gelatin. So all these different types of materials are possible. In our lab, we have specialized in developing materials from tissue. So what we do, basically we collect the tissue from the slaughterhouse or even from the hospital, and then we collect that tissue, we throw out all the cells from that tissue, and then we develop the acellular matrix or the extracellular matrix. And that matrix, it has most of the component that is present in our tissue. Okay, so this is a very much biomimetic material that we develop, and then we can load the cells within this biomimetic material, so the cells could survive there, and then we print them into 3D structures with the help of our 3D bioprinter. And then basically the 3D printed structure, we mature them, and that can be used for different applications like whatever I have earlier shown you. Okay, so little bit details about this work. This is from our own work, where we have developed the tissue, like we have developed this hydrogel from 3 tissues at least: cartilage tissue, heart tissue, and adipose tissue. And nowadays we are working on many other tissues also. And then these tissues are desolarized. Desolarized process is like removing the cells from the matrix. So the cells are gone. So cells are the mostly cells are the immunogenic factors that is present in the tissue. So that's why the whole tissue cannot be used to implant in other body. But if you remove the cells, if you can wash the cells in a very good way, so that in the tissue, if the cells that is not there, that then the tissue material, that the extracellular matrix, that is not immunogenic in nature. Now from the studies, it has been found that. So that's why this destroyed extracellular matrix we have developed like this way. And then we have also characterized them by various ways, like either by the DNA quantification, collagen estimation, and GAG estimation. And those are very important to do that, because by DNA estimation we can understand how much amount of cells are removed, what is the content of the cells removed, or what is the content of cellular material retained. And by the collagen and GAG assay, we understand that how much amount of the material is retained within the desolarized tissue. Now, when you do that, when you prepare the hydrogel from the tissue, it is a very good material, because now this is at 15 degrees centigrade, this gel can move, or it can flow basically. So it's flowable at 15 degrees centigrade, but it behaves like a gel at 37 degrees centigrade. So now we found a very good way for our printing. So for our printing, we need to have the temperature around 15 degrees centigrade. And then, so after printing, if we kept them at 37 degrees centigrade, they will become gel. So that's without any further other cross-linking, cross linkers, we can able to make them gel by thermal gelation making. And then we have also characterized them by various rheological properties like that. We have measured the shear viscosity of the material, and from this viscosity curve, anybody can tell that this is a shear thinning material. And then at 4 degrees centigrade, you see that the modulus is much lower, but at 37 degrees, the modulus is going higher. So from this curve also, we know that the material will become gel at 37 degrees centigrade. And then also after gelation, there is the storage modulus and loss modulus study. This is the way we have printed them. You can also print the gel only the gel can also be printed, but we will not able to get a very good structure from that. So we mixed this. So what we did, we printed some along with the thermoplastic polymer, like PCL is thermoplastic polymer. So when we printed the PCL framework, then in these lines, in the gaps, we printed the hydrogel or the DCM hydrogel, destabilized extracellular matrix hydrogel. We have printed in between the layers, so you can get a hybrid structure. This is the video of this from our study where we have done what we have done. We have printed first the PCL framework, that is the thermoplastic polymer. When the PCL framework is first printed in a layer in a line by line manner, so we are keeping the gaps in between the lines. Now this process is repeated to get the and then the same hydrogel, and where cells are also mixed, cells are also loaded here, and then we can make we can load them, we can print them basically in the gap between the line. In this case, we have used two different types of hydrogel because we have shown the possibility of using two types of hydrogel at least in a single construct, so that in many times you know that suppose your tissue is a composite tissue, then we need to have different types of bioinfinity, we need to print different types of cells. So that's our interest, so we can print the structure. Then we have done various in vitro assays, like how we have cultured this tissue, like we have cultured the tissue in an incubator, CO2 incubator, and then we have seen that, like here, this is the cell-laden structure that I was talking about, where this is shown under a bright field microscope, where the cells are visible also. Here, this is the same microscope of the same thing. Here, they also this bright field phase contrast micrographs of the same thing. Where these black lines are showing, this is the PCL line that is printed, that is the thermoplastic polymer, and also in between the lines, this hydrogel are printed, where the hydrogel, where the cells, these round things are the cells that is also loaded into this thing, right? So this is the hybrid structure or the cell printed 3D bioprinted construct that we have done. And now the next thing is to understand whether the hydrogel is present everywhere or not, and whether the cells are viable within this construct or not. So this is the how we have done cell viability study, where the cell, all the green dots, those are the live cells that is present inside the structure, and it is almost present everywhere. And then the structure is also stable till 14 days. That's what we have analyzed, but it can be stable. It is stable more than that also, that we found later on. Now, one primary concern of this 3D bioprinting, extrusion-based bioprinting technology is that it can, because you know that when you are pushing a material through a very fine orifice, there can be a shear stress generated, right? So that shear stress can induce cell apoptosis or cell death. So you need to find out that whether our system, when you are doing bioprinting, that system is imparting any cell death or not. So that's what we have. Tunnel assay we have done, and tunnel assay is a very good assay to identify whether the cells are apoptotic or not. But we have found that printing and non-printing, there is no big difference, so that the cells are not apoptotic, or that is by this process that cells are not getting apoptosis. Next thing is also we have analyzed the cell viability, because our interest is we try to understand, try to see whether what is the level of cell viability, or how many cells are viable. And you see here, all the green dot things are the live cells, and there are red dots are present. Those are the dead cells, right? And almost more than 90% cells are viable within the till 14 days that we have measured. Another important thing is that these cells can connect to each other three-dimensionally. This cell-to-cell connection is very much important for tissue building, because unless the cell-cell interaction is established, the tissue building will not take place. That's what we have found. Then also we have done various other things like the gene and protein expression assay, where we try to understand what types of genes are basically expressed, right? Because each tissue, they have their specific genes. And after the tissue printing, we would also like to see whether what types of genes genetic expressions are there, and what types of proteins are also expressed by the tissue, because the protein expresses the function of that particular tissue. So that tissue, suppose if it is a bone tissue, that should be able to produce collagen 1. Skin tissue also should be able to produce collagen 1. If it is cartilage tissue, should be able to produce collagen 2. So that's what we have seen here. So whether this tissue is doing the function that is like that, that's very specific for that particular tissue or not. I'm going to discuss a different example where we have also tried to print a complex construct. Now, if you have a very complex tissue, suppose that is not a very simple skin-like two-dimensional structures. You have a complex tissue. So suppose if your interest is to develop this human ear model. So basically, we'll start with the CT data. From the CT, we'll get the CAD model. We can generate the CAD model of that ear, and then from that one, we can generate the virtual blueprint of this ear model. Then we can, with the help of this bioprinter, we can able to print this structure. Now, there is one challenge here. In the ear, you see there are a lot of this geometry here. Those are hanging. Either you can print it on the reverse or anywhere. If you print them, they are a hanging geometry, and that cannot be printed without any support. So for that, you need to have a support structure. For that, what we have done, we have used the sacrificial material here. So this yellow color thing is the sacrificial material, and the white color thing, that is the structural material. Now we have printed both simultaneously in every layers, so that we can able to, suppose our interest is to print this kind of reverse pyramid structure. So where these two faces, where it is hanging, right? So we were able to put the support structure. So that's what we have done. We put the support structure, and then after the printing is done, we can just basically remove the support just by putting them in water or media or buffer, because this sacrificial material is also but this is a water soluble material. Like in this case, we have used PEG, so that you can easily remove the supporting material or sacrificial material, and we can get the desired structure. So with this help of this technology, we have developed this kind of ear model structure where this is the mostly it is a chondrocytes, the pinna, and then the ear lobe, that is mostly fat tissue. So we have printed the cartilage cells here and the fat cells here, and that is why we have printed the whole structure. Now I'm going to show you another example of this 3D bioprinting, where this 3D printing technology can also be used for facial reconstruction or for maxillofacial implants. How we can actually, suppose if you have somebody has a trauma or defect anywhere in the facial or the maxillofacial region, so that then it is a major it is a social issue for them. He will not be able to go out. He will not be able to mingle with people. That is a major issue, right? So how we able to do that? Another thing is, now suppose somebody has a cancer in some region, and that cancer was because to remove the cancer from some part of the tissue is resected out. How we can do that? Suppose this boy, he had a cancer, and below his orbital cavity here, and that part of the bone is taken out, right, to remove the cancer. So when we got the CT data, we identified that this is the region here. That is the missing part. He doesn't have that part, basically, because that part was resected out. Now we all have across our this midline, we have symmetry to left and right side. We have a symmetry, right? So if we can just superimpose this left side and right side, we will be able to get this, the missing part. So this is the missing part that was taken out. So you know that now this is the structure we should be able to 3D print. So we have done that. Basically, we have developed the CAD model. We have printed the structure, like this structure we have printed with the PCL, and then we have given to the surgeon who has done the surgery. So he basically put those things in the patient, that defect area, and sutured it back. And then this is the image after 4 days, and this is after 2 weeks. You see this now, his eyes are put again aligned back, and also now he is much more confident. He can go out. He doesn't have this social stigma, and he's feeling much better now, right? So this kind of maxillofacial defect or defect due to trauma or due to cancer, that can be well rectified by this kind of 3D printing technology. In vitro model development. And why? And it is like starting with the lead molecules, then passing through the 2D cell culture test, and then few lead molecules will get passed through. Those are not toxic. And then testing them on a small small animal model, then large animal model, then volunteers, and then ultimately finally for the patients, right? So this process is very long, a lot of money is required, and also many times these this screening platforms or screening models like the 2D culture model, animal model, they are not beneficial. They can give false positive or false negative result. Other thing is that the drug may be working well on the animal, but maybe the drug is not good for animal use, right? So that kind of we cannot identify from this test. So now that's why there is a tremendous amount of interest on this developing these in vitro models, where these models can be uh, well used for screening the drugs at the beginning itself. So we can use the human-specific models at the initial stage itself, so that we can identify the lead molecule that is human-specific, and so that we can identify the molecule at the very beginning, and then we can do other tests. And recently, as per the different this regulatory bodies like the EU or others, they banned all animal-tested products. Okay, basically for cosmetics, they have definitely banned, and for other also now that is going to do soon. So we need to then, that's why there is a tremendous amount of interest to develop different 3D models that can be well used for this drug discovery or drug screening purposes. And the advantage of all these tissue models are that can be like we can do systematic study also. We can repeatedly use them repetitively. They are very high throughput, and quantitative investigation is possible because we can just change the dose and see what dose that is their response is there. Also, we can also test them at very low dose because these tissue models they are very fine, very minute, like very small, so that the drug requirement or the media requirement is very low. So that's why we can do this test very quickly. And combination of drugs molecules can also be used. You can understand mechanism of action and dosimetry, like what is the different doses. That's what we can do. One of the major research interests is to develop in vitro liver model. You know that the liver is the primary organ in our body that does most of the metabolism in our body. So whatever the drugs we are taking through oral route, like tablets, capsules, whatever, the drugs that is mostly it is going through the liver, right? The liver is the organ that there it is detoxifying the body. Also, it is metabolizing the drug, right? So that's why liver is a main primary research interest for many researchers, because they try to develop the liver model, and that model can be well used for this drug screening. And if you know that liver is an organ, like it has millions of lobules. Lobules are this kind of hexagonal units where it is like the blood is flowing through one end, the terminal end to the central end, and then the hepatocytes is sitting just beside these channels where the hepatocytes is basically taking out the oxygen and nutrients. And because this hepatocyte, they are metabolically very active cells, so they need high amount of oxygen and this thing. That's why this kind of architecture is there. Now, can we generate this kind of architecture in vitro setup so that we will be also able to understand the drug interaction through this liver and other thing? So we are working in that direction, but I have not included that thing because our work is a little bit premature where we have not published that work yet. So that's why. But this is a work from an industry like Organovo is a bioprinting industry. It's a bioprinting industry in US, and they have come up with a in vitro liver model in a well-plated platform. That is a 96 well. They can print the liver tissue, and that can be used for drug screening. So they are selling this thing to the pharma industry, and this is a video from their website. I have taken this video from their website. Like what they are doing here, basically they have used this extracellular bioprinting technique for printing the liver tissue in a well plate. This is a 96 well plate, where in each well plate it is basically print a small liver tissue, micro liver tissue you can print. Okay, and with the help of this hepatocytes and endothelial cells, you can print. So these are the hepatocyte island, and you see these hepatocyte cells, they can produce the small tiny blood vessels within the printed construct. So this looks like this is a very biomimetic liver tissue. And then if you add drugs, if the drug is toxic, then the all cells will die, right? So that's the test that can be easily done. So like you can test whether the cells are viable within the construct or not. And then if you have some hepatoprotective drug, and that so that it can again put the cells back viable. And it is better than the two-cell culture because here the three-dimensional structure is important. And in our tissue, it is a three-dimensional tissue, the cell interaction and cell-matrix interaction and three-dimensional are very much important. So unless we have this three-dimensional tissue structure, we will not be able to identify the drug response to this tissue that happens in our body. So that if you want to get a drug response that is very much important or very much mimicking or very much similar to our drug response to our body system. So that's why we need this kind of three-dimensional bioprinted models are very much important. So this is one thing. So now there are various other types of 3D bioprinted models research are going on, like 3D bioprinted skin models, kidney models, and various other tissues. So if you search that, I have not included those things here because due to the time limitation, I have just included one example, but there are many examples of each of these cases, and I would request you to go through please search for that you will be able to find many more information from for these things. And the in vitro application of this technology, like in vitro liver model, can be many things, as many other things. We can understand the drug screening is very much possible. When we can actually how this infection actually spread, thing can be understood. I mean, that can be studied in vitro setup with the help of this kind of 3D bioprinting technology. And in vitro disease model, cancer model can also be used if you use that kind of appropriate cells. And if you use patient cells, cancer from that will reproduce, that thing can be done, right? Now I almost came up presentation. This is the last slide where I'm going to discuss about the very important questions, or many of you probably have this question. So when use they readily available, they are not available in clinics. So, but that's why the cautionary statement is till now no bioprinted organ is put into human clinical trial yet, because mostly they are in the lab or in their or the various in vitro or various cancers, sorry, not cancer, various studies, studies are going on, and many of them are taking a couple of years. Technical challenges. Both we have to. So in two-dimensional thing, like two-dimensional tissues organ like skin, cartilage, those are very much possible in short term, but if they are like hollow tips or hollow organs, those are required, those can be possible in mid-term, and the solid organs that can be possible in long term. Something like very soon, we may probably in the next couple of years, we'll be able to get joint cartilage, but will this skin, peripheral nerve. So these things are possible to get in the next 2 to 5 years, but some little bit complex tissue like trachea, throat cartilage, heart valves, anterior cruciate ligaments, these things may be possible in another 10 years down the line. Okay, and then if you were thinking of solid organs like kidney failure, liver failure, complex organ failure, so those will take a little more time, because it may take maybe 10-20 years before they can be available to patients for implantation purpose. Now here there are major challenges here why it is so. One thing is like this multidisciplinary approach. So here we need participation of different people from different backgrounds. People from mechanical engineering, structural engineering, design, people from, and on the science background, people from science like human anatomy, physiology. On the clinical background, people like doctors, surgeons, and also biomedical engineers, and of course the regulatory agencies. So we need participation of all different kinds of people to develop this to develop further this technology into a commercially viable technology where we can actually print the different tissues or organs that can be well used for implantation purpose. So definitely one day that day will come when if somebody is somebody has a organ failure, let's suppose major organ failure like a liver or kidney, then the doctor can able to say that you can wait here for a few hours. Your liver or this kidney is getting printed and it will be implanted on you soon. So because there is if you see here, there is a huge disparity or difference between the demand and supply. The demand is much higher than the supply because organ donation is not that popular, and organ donation we cannot get the organ, and there are many other challenges also. So that's why if we can develop this technology into uh, a life-saving technology, that would be the ultimate goal of this bioprinting community also. So let me acknowledge many people here because, as I tell, this is a very multidisciplinary approach, and in our team also, we have a multidisciplinary team here at IIT Hyderabad. There are many people working on different aspects of this technology, and also I'm fortunate to get a few grants from different funding agencies like DSTSR, DBT, and MHRD, ICMR. Thanks to them, because this area is this area needs huge amount of resources, and funding is very much required for that. And also we have established collaborations with different academic institutions or with different scientists in abroad, and also as well as in India, and basically and also in Hyderabad. I have done that. And now also we have a industry collaborator who is also giving some amount of funding for our one proper project. And these are the people, they are very important because they are the ones who actually doing all the work in the laboratory. Okay, so that I can able to present the data, I can able to speak in front of you and show our research because of them. So thanks to all of them, these are the PhD students and master students, those who are working hard in various topics of this related to 3D bioprinting. And also, in conclusion, I would like to say that though organ printing is very challenging, and it needs participation of many people. It needs expertise from different disciplines, and it is very complex because organs are very complex. You know that because our tissues organ that developed over a billion years of evolution. Now, with the span of a few years, we are trying to develop something. So that's a very challenging thing. So that's what. But anyway, organ printing is very highly huge. It has a huge prospect. It can do many more things, and many more things are possible. Not everything is done, and this is a very nascent technology. But one thing is, don't lose hope. The future is not that far away. Definitely, we'll reach one day when we'll be able to print any tissues or organs that can save a patient, or that can be useful to treat certain disease or certain ailments. Thank you very much, all of you, for joining me here and for patient listening. And I'll be happy to answer if you have any question, comments. I'll be happy to answer that. And my email ID is here by falguni@bme.ith.ac.in. You are welcome to write to me if you want to get associated with us to work in this beautiful discipline. And also, if you have any question, comments, and you are not able to ask today, you definitely can drop me an email, and I will be definitely try to answer you. And if you want to collaborate with me for any of this on any of our research endeavors, I'll be more than happy to collaborate. Thank you very much.
Thank you, sir. Thank you for your wonderful speech, sir. That was quite interesting and knowledgeable. I think most of the participants were having doubts related to the 3D printing and bioprinting. So their doubts will going to clear what is exactly 3D printing and what is exactly the bioprinting.
Yes.
So I request all the participants to unmute yourself and ask questions if you have any queries or if you want to discuss anything with the speaker. Undergo research only an inside body and will promote bone in growth.
Sorry, if I understood, if there is a 3D printable material for bone tissue, right? That's what.
Bone tissue, sir.
Okay.
That bone will
Actually,
growth inside the body or not?
Yes. No, actually, for bioprinting, the aim is that whatever we are printing, that is biodegradable. Okay, so whatever material we are using here, they are biodegradable. So we don't use any metal or metallic material here. Okay, so what you are putting, that will be degradable. And in a way, what it does, it helps the cells to grow. So the cell number will increase. The cells will produce the extracellular matrix. So the natural bone will get formed in that area whenever we are putting this material. So that's why there are various types of materials that can be used for bone printing. Like we are using collagen, chitosense, gelatin. Also, another thing we are also doing, we are also using some kind of thermoplastic materials like PCl along with tricalcium phosphate or hydroxyapatite. Those are also being used for printing this bone tissue.
Thank you very much, sir, for such insightful session. Sir, can I know during organ printing, what if different cell tissues are required? There is question from Mr. Salman Shah Khadri.
Okay. Yeah. Hello. So when you are doing a particular tissue, 3D printing for a particular tissue or organ, first we see what is the target tissue. Okay. Suppose if you are trying to print skin, then we see what is the morphology or what is the anatomy of the skin, what are the cells located at different layers, or different how the cellular organization is there, how the cells are ordered in that particular. We try to understand that first. So taking clue from there, then we try to print the similar cells. Like if you are going for skin printing, then we'll be using fibroblast cells, that is the resident cells of the skin, and keratinocyte cells, and other cells also. Then we'll print the fibroblasts as they are positioned in the skin. We will to print them like in a particular order. And then the next cells will be also printing on top of that in that particular order, in a particular order. So basically, we are trying to mimic the tissue that we are trying to print while going for this by printing.
So depends on our requirement application of a particular body part, we are going to use the different kind of cells.
Different types of cells. For bone we will be using osteoblast. For cartilage, we will be using chondrocytes. Similar to for different tissue or organ, different types of cells are being used. Also, we can use stem cells, and then we can differentiate them to a particular lineages.
Is there any questions?
Sir, what about the cost of the organs printed by the 3D printer?
You see, the cost will not be a matter here, because you see this is a life-saving technology. So suppose if somebody can give you a printed liver, so you will not be this thing about the cost. But coming to the cost here, the cost will be now, because this technology is very new now. Now the cost will be probably a little more, okay. But as we progress much, as we develop newer technology, as we develop newer materials, or if the technology progress further, then the cost will come down. Okay.
Okay.
So there is question from Jenny. To hold the cells, it needs matrix. Example, bone.
Yes.
It needs matrix and cells together. The matrix contains inorganic and organic materials. So matrix needs to be fused. Then how to print cells and matrix together? I'm mentioning about reconstructing a large bony part. She's talking about bony part, large bony part.
Yeah, I understood that. That's a very good question. Actually, when the matrix requirement is typically there differently, and when we printing, suppose printing bone cells, okay. So what we do, we mix the bone cells in a soft material like collagen or any other thing, right? But with that soft material, we will not be able to print a bone. Well, bone is much harder material. So what you do, as I shown you one thing, that hybrid printing concept, where we print suppose we print with PCl, we print the framework of the bone, and in between the gaps, we can print this hydrogel containing bone cells. We can print in between. So the thermoplastic material will able to bear the load that is required for this bone tissue, but the cells will be encapsulated in the soft material or the hydrogel, so the cells could thrive. Because with the PCl, we cannot put the cells. So that's why for bone printing, actually we use in our lab. There is a maxillofacial surgeon who is doing PhD with us. So he is working on this thing. So she has developed a hydrogel, and she has also developed the material where we are trying to do this hybrid bioprinting concept to print a bone cell for a bone tissue. And there actually we take the CT data from the patient of the defect, so that we can understand, we can exactly match the shape and size of that requirement of the defect, so that we will be able to print the structure that will exactly fit to that particular area.
Thanks, sir. Thanks for your answering the questions. Is there any questions? Any questions?
Sir, will you share the life assessment of the organs?
Sorry?
Sir, will you share the life assessment of the organs printed by the 3D printer?
Life assessment, right?
Yes.
You're saying, right? Yeah. No, life assessment is like, as I said you, like these organs, they are meant to be implanted, right? So if
Okay.
they can integrate with the body, then there is no problem.
Okay.
So then they can be there for the throughout the patient's life. Okay. But that integration is very important. So the major important, the integration of this bioprinted organ with the patient's body. Okay. That
Okay.
is, so we are doing various animal testing, and from the animal testing we could understand that yes, these things, these bioprinted tissues are getting integrated with the body. Okay, with the animal body, so there is no problem as such. But it is like in future we need to wait further, so to understand whether how these bioprinted tissues will react in the patient's body.
Yes. Yes. Okay. 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 Thank you, sir.
Yeah.
There is another question from Dr. Jenny. Can we say seed stem cells to a printed pig scaffold and promote growth? Which is best material to promote vascular ingrowth and cell migration?
Okay, so peak is a very good material to use now. Nowadays, many people are using peak for 3D printing of structure. And yes, we can load the cells, or we can seed the cells on the structure. So that peak can be used as a scaffold basically. So that's not actually bioprinting, but what we do, that with the peak we can 3D print a bone scaffold, and on that we can load the cells, or we can seed the cells on the structure that can be integrated, that can be implanted in the patient's body, and then implanted in the in vivo system. Okay. But for if you're asking me like what are the good material for bone vascularization, right? That is the another question, right?
You can answer that.
Sir, yes, sir. Yes, sir.
Yeah. So like for vascularization, for inducing vascularization, generally we use different types of things. Like there are like some growth factors like VEGF and other stuff that those are being used. Also in certain areas, certain other things like there are some pro-angiogenic nanoparticles, something like europium hydroxide or any other zinc hydroxide or zinc oxide or those such. So these materials can also be incorporated within this bone scaffold, and then they can promote the vascularization. I don't know if I can answer that too.
Yes, Dr. Jenny, if you have any doubt, you can ask the speaker. Dr. Jenny? Okay. So we have another question, sir. What is the difference of having titanium alloy and bioink printed parts in a human body? If suppose what a patient will going to feel or long term in short term as you have
Yes.
described that. So
Yeah.
what a patient or what are the issues will be facing by a patient if we implant titanium alloy object, human body part and a bioink printed object
Yes.
body part?
Yes. Yeah. No, that's a very, very good question. The major difference is the degradability. Titanium implant is not degradable, so it is going to stay throughout the patient's life. It's not going to degrade. So it's a biostable. Okay, biostable income. So that the titanium implant, when you are making you're putting into the patient or implanting that or putting that in the body, so that is going to stay for lifelong. Okay, and there are a few cases there can be like the tissue can grow over this titanium implant. It has been seen that the tissue can grow or integrate with the titanium implant. Okay, but there can be a mechanical mismatch, so that can cause problem later on in the down the line. That can be there. But with the bioprinted organ or tissue, the beauty is that these are biocompatible, these are biodegradable, the most major thing. So it is not going to stay in your body for long. But what it does, it helps or facilitates the tissue regeneration. So the body's the native tissue or the natural tissue will come to its place. Basically, you are using our body's regeneration potential with the help of a bioprinted organ or tissue. So that in that way, so down the line, this bioprinted organ or tissue, it is not going to be there, and basically your body will develop its own tissue in that place. So you are using your regeneration power with the help of this thing. So that is the major difference. So after some time, you will not feel a difference between the bioprinted tissue and your own tissue.
Thanks, sir. There is another question from Dr. Jenny, sir. How to incorporate proteins like bone morphogenic proteins in 3D printed peak or polymers that may get damaged at sintering temperature?
Yes, that is the one major challenge of using ceramic, incorporating growth factors or any other this kind of bone morphogenic proteins. Those are highly temperature sensitive material. So what we can do, like after sintering, you can incorporate them by some technique. Okay. So that's basically people do it like this. So you can use the various kind of chemistry. Either you can use various type of chemistry, or you can use some kind of hydrogel or something to integrate your bone morphogenic protein with the ceramic structure, or even with the peak structure. You can do that. But basically, you need to integrate the bone morphogenic protein after the printing and after the sintering is done.
Sir, there is another question. Can we print or modify the kidney for solving kidney-related problems using bioprinting technology?
Yeah, actually, there is a many research groups are working in that direction. Because kidney is a major organ that does all the things, and there are many patients suffering from this thing. You know that there are many of them. They are undergoing dialysis. There is kidney damage. All these things are there. So definitely, there is a huge need to do this 3D bioprinting of kidney. But as I already warned you, like this is not very easy to do that. There is a huge, like this is a very complex organ to bioprint that, but definitely it is required, and there a huge amount of research is going in that direction. And probably one day we'll be able to do that. We're able to bioprint a kidney, suppose, for this purpose, for to help the patient who are suffering from this kidney failure or this kind of thing.
Thanks, sir. Is there any questions?
If you cannot ask now, we can also drop a mail to me. I'll be happy to answer, sir, if I can.
Sir is very kind to answer your questions through mail also. You can ask if you have any doubts. So we will going to conclude, sir. Sir, I once again thank you for giving a wonderful lecture that was interesting, and from all the lectures which we have seen from 4th January from this FDP, this session is totally different, sir, as it is related to the bioprinting. So once again, I like to highlight, sir, we have discussed about the bioprinting methods, inkjet, extrusion, laser assisted methods, and one of the video you have shown that skin defect, how to recover the skin defect problem using that video. You have that video was very interesting, sir, and various process parameters the bioprinting and the gel bioink also, sir. 3D printing in gel and the various human body parts and the challenges of printing that human parts directly, like USA have done the printing of the human heart. That was also quite interesting, sir. And you have also discussed about the various properties of the bioinks functions and how we can print the ears and the liver tissues. Thanks for this session, sir. That was purely knowledgeable, technical, and resourceful, sir.
Thank
Once
you.
again,
Thank
I
you
once
very
again
much for
I
Thank you
once
very
again
much for giving me the opportunity.
thank Dr. Falguni Pati, Associate Professor, Department of Biomedical Engineering from IIT Hyderabad, for giving a wonderful lecture on recent trends in bio printing for biomedical applications. Thanks, sir. Thanks once again.
Thank you very much.